Okay, did a trial run. This should work. I'm working on specific defenses now, and we're going to review the material about the specific defenses, and we'll use the figures from the other book just to give you a different view of things. I wanted to start out by reminding you that the specific defenses are the things that are constituting the immune response, and that there's two primary avenues. One is the cell-mediated, in which you get your T cells that are going to directly attack cells that are infected or the pathogens. And the other route is through the B cells, in which the B cells are going to produce antibodies, and then the antibodies are going to label the antigens and can either then work with the nonspecific defenses or with the T cells in order to get rid of them altogether. Okay, more about that later. Oops, what did I do? I don't know. Keep going. All right, so this is... let me put that down. Okay, antigen presentation on the MHC1 protein. Okay, so any cell that's infected can have these MHC1 proteins, which we're going to show right here. Okay, so these MHC1 proteins, what's going to happen with these MHC1 proteins is these MHC1 proteins are going to sort of continually migrate through the Golgi, through the vesicles, up to the cell surface. And sometimes they're going to either be empty or they're going to have just like normal peptide stuff. But if they happen to be infected, there's going to be certain abnormal peptides, which we consider to be antigens, present in that MHC1 protein, so that when it's popped up onto the surface, it's actually showing up the antigen. Okay, so the analogy that I used was that the MHC1 protein is going to be the flagpole, and the flag then is going to be the antigen. So keep in mind that these MHC1 proteins are continually being moved up to the surface. Uninfected cells will not show antigens. Infected cells will show antigens. Uninfected cells will just have a flagpole, meaning an MHC1 protein, but no antigen, no flag. Okay? The next version is the MHC II proteins, which are only present on antigen-presenting cells. Okay, now, antigen-presenting cells are going to be phagocytic cells, like macrophages. And what they do is they will engulf, through phagocytosis, the pathogen, break it up into a bunch of little pieces, and then present that on MHC2 proteins. Okay, so MHC2 proteins are going to be found on antigen-presenting cells. The only time that you see the MHC2 proteins is going to be when there is antigen present. If there is no antigen, there's no MHC2 protein on the surface of the cell. Okay? So, you can see MHC1 proteins on the surface of any cell that has a nucleus. It may contain an antigen. In that case, the cell is infected and it will be marked for destruction, or it may be empty or with normal stuff, and that way it's disregarded. MHC2 proteins are only going to be found on antigen-presenting cells, and they're only going to be found on the surface of the cell if, in fact, that cell has antigen to display. Okay, now these MHC proteins are going to enable the T cells to recognize them, activate them. Okay, so antigen presentation was the first step. The next step is going to be recognition. So now we've got to recognize that antigen on the MHC1 protein. So we're going to start with the MHC1 proteins. Those MHC1 proteins are going to be recognized by cytotoxic T cells, and those cytotoxic T cells are going to become activated, and the activation of the cytotoxic T cells will lead to the destruction of the infected cell. Okay, so this is how it's done. Okay, so first of all, let's review the fact that the cytotoxic T cells are going to have a marker on their cell surface called a CD8 marker. And as they show here, they're trying to show that that CD8 marker is going to help with the recognition of the cytotoxic T cell and the MHC protein and the antigen. So it just kind of helps with this complex formation. Now, what's got to happen here is that this infected cell is going to interact with a cytotoxic T cell that's going to have a receptor. This T cell receptor right here is going to be specific for that antigen as well. Okay, so this cytotoxic T cell receptor is specific for that antigen. And so once you get this sort of binding going on, there's that co-stimulation thing, which I said is just a double check process that you don't need to worry a lot about. That's going to lead to the activation of that cytotoxic T cell. Okay, so once this binding here takes place, that's going to say, oh, okay, we've got the right antigen. Let's make a whole bunch more of that same exact cytotoxic T cell, and let's send these cells out to go looking for more of those antigens. And let's also make a couple extra and tuck them away to live in the reserve section. Okay, so now these active ones are going to head out into the bloodstream. They're going to check out all the lymphatic organs, and they're going to look for more cells that are infected. And once they are, they're going to find different ways of trying to destroy them. And I just realized that this says some different ways, but basically the ideas are the same. Perforin is one of the things that is going to lead to the destruction of the cell membrane. Now, let's just remember that perforin is going to work with granzymes, and that those are going to stimulate apoptosis. And cytokines are also going to stimulate the apoptosis, and there's some other toxins that can be stop. Okay, so I just checked with the other one just to get a reminder of what the other book says. So the other book does mention the the perforin, and it mentions tumor necrosis factor and interleukins. So just keep in mind, I mean, cytokines are the same thing as interleukins, and tumor necrosis factor, and basically the idea is that we can also get macrophages coming to destroy the cell. But basically the idea is, and the goal is, to destroy that target cell, the cell that's infected. So what's going to happen then, this is just a little review. The MHC1 proteins can be on any cell that's infected or somehow abnormal that's going to express those antigens, that it's going to be recognized by those CD8 T cells, which are the same thing as the cytotoxic T cells, that the cytotoxic T cells are going to attack and destroy. We're also going to produce some extra memory cells, and let's not worry about the suppressor cells for now. Okay, next we want to see the recognition of the MHC II proteins, and those are going to be acting by the helper T cells. And so in this case, we've got an antigen-presenting cell. So remember, the antigen-presenting cell is now presenting the antigen on an MHC2 protein. That MHC2 protein is only going to be present on the surface of the cell if the cell has antigen to display. And so it's going to be recognized then by the helper T cell, the CD4 cells. And this is just kind of showing how that they're going to interact together. Keep in mind that helper T cell is going to have a receptor specific for that antigen. Once these bind together, they're going to activate those helper T cells, and you're going to get that mitotic division thing. The current book is going to call that mitotic division thing clonal selection. And that clonal selection process is basically when you make a whole bunch of cells that are exactly the same thing that we did for the cytotoxic T cells. And those cells are then going to release the cytokines, and by releasing those cytokines, that's how the helper T cells are going to coordinate all of the different types of defenses. Okay. So this one is going to break down and show how it's activating the B cells. So the thing about the B cell is that the B cells are going to interact with an activated helper T cell, and the B cell is going to have receptors that are going to be specific for that same antigen again. And you don't need to get the whole process down here. Just keep in mind the fact that the B cell is going to be specific for that particular antigen. It's going to interact with an activated helper T cell, and that's going to stimulate this B cell to transform itself into one of these plasma cells, and these plasma cells are going to produce lots and lots and lots of antibodies. Okay, also keep in mind along the way we're going to make some extras and keep them as memory cells, and those are going to be the reserves. Okay, so the summary for this is that the MHC2s are going to be on antigen-presenting cells only, and they are going to always be indicating that you've got some sort of infection. And the helper T cells are going to stimulate the immune response by the T cells, the B cells, and also it's going to attract the NK cells.